All right, now that I'm not on mute, we're going to get started. So when we left off on Monday, we were talking about acid-base balance, and we had kind of gone back to the respiratory system to look at the influence of carbon dioxide. Today, we're going to go back to the kidneys. So we're going to talk for a short second about the interaction between fluids and electrolytes. So returning to our solutes and water before we move on to the different factors in the renal system that affect our acid-base balance. Okay. Okay. So these are the last two sections of chapter 19. Hopefully we'll get through them today, and there will be no class on Friday. Okay. So we're starting with fluid and electrolytes. So we've talked about each of our substances sort of in isolation. We talked about how water gets reabsorbed. We talked about how sodium and potassium get reabsorbed or excreted. And we talked about specific hormones kind of independently, but this is basically just a note to tell you that all of these things are happening at once, and actually most of these things have multiple effects. So whenever we're thinking about solute reabsorption, what we really want to be thinking about, and what I've tried to emphasize as we went through it, is that really one of the really important parts of that is that increasing solute reabsorption is increasing the osmotic gradient for water reabsorption, which is kind of the point of increasing your solute reabsorption, is so that you can maintain appropriate blood volumes. These hormones do have some other actions in addition to the ones we went through as we went through each of our solutes. So ADH, which we talked about when we were talking about its role in water balance, because it can insert aquaporin 2 into the apical membrane to make the distal tubules and collecting ducts permeable to water. It actually also increases the number of sodium channels in the apical membrane of principal cells. So it's letting water get reabsorbed, but it's also going to influence our solute reabsorption, because at the same time as it's letting in water, it's going to let in sodium, because these two things kind of move together. So angiotensin 2, we talked about for its role in stimulating the secretion of aldosterone. We were talking about aldosterone as we talked about sodium and potassium, but angiotensin 2 also increases ADH secretion, which we kind of saw in our flow chart for angiotensin 2 in one of the earlier slides in the last set of slides. So in addition to having us reabsorb more sodium, it also increases ADH secretion, so it also increases our water reabsorption. ANP, in addition to decreasing the amount of sodium we're reabsorbing, it also decreases ADH secretion. So in addition to not reabsorbing solutes, so not having this osmotic gradient to pull water, it also kind of actively stops us from inserting those aquaporins in the apical membrane. So it's stopping that reabsorption of water kind of in two ways. So each of these have multiple roles other than the primary one that we went through as we were talking about electrolyte balance. Okay. The point of having these interactions between the electrolyte balance and our fluid balance is so that we can respond to physiologic or biological events, specifically things that are going to happen to your body and happen to your cardiovascular system, like hemorrhaging. So in a hemorrhage, you decrease the amount of blood volume because you're bleeding out. So you decrease your mean arterial pressure. And you're going to have a bunch of different responses to that, right? So you'll have neural control of your heart, of your cardiovascular system more broadly, including our baroreceptor reflex. So that reflex that basically upregulates our sympathetic nervous system to try and control our blood pressure from that way. But we're also going to have a hormonal response in response to this decrease in mean arterial pressure. So if we're kind of trying to tie everything together that would respond to a decrease in mean arterial pressure, we want to think about both our fluid and our solutes. So we're going to think about both ADH and our renin-angiotensin-aldosterone system. So we think of ADH primarily when we're thinking about water. We think about the renin-angiotensin-aldosterone system primarily when we're thinking about solutes. But both water and reabsorption of solutes are a response to decrease in mean arterial pressure because they're both going to help us conserve plasma volume. Okay. So that's just kind of tying it together, and hopefully we talked about that. Really, I tried to emphasize it as we were talking about solutes and water. We can go through more of that a little later as we review for the exam. But I want to get back to acid-base balance because that's really what our last topic for this chapter, for this unit is. So we talked about acid-base balance in a brief overview when we were talking about how it is linked into the respiratory system when we talk on Monday. So we saw that we have a normal pH of arterial blood, and that normal pH is under homeostatic control. We want to keep it in a specific range, which is about 7.35 to 7.45 pH. So it's slightly alkaline here. If we have a lower pH, lower than 7.35, so lower than that regulated level, we're calling it acidosis. We have a higher pH than that regulated level, we're calling it alkalosis. When we're thinking about the pH of blood, really what we're thinking about is the presence of hydrogen ions and the things that buffer them, so bicarbonate primarily. And we'll be talking about bicarbonate a lot now. We mainly focused on biologic acids like carbon dioxide on Monday, which will play a role in this. But now we're going to kind of talk about how the kidneys can compensate for that. Okay. But just a reminder, the complications with a disturbance to our acid-base balance, so a change from our normal arterial blood pH, are really due to the fact that they change protein structure. When you have a different concentration of hydrogen ions floating around, it messes with the shape of your proteins, which messes with their functions in all sorts of different ways, depending on what proteins they are. And honestly, we don't really know all the exact specifications of what's changing and how. Mostly we know that it causes really big problems and even death. But some of the observable things that we can observe with acid-base disturbances is that our neurons get more excitable in the central nervous system in alkalosis, whereas they get kind of depressed in acidosis. And so we can see this in kind of the actions of your nerves themselves. So if you start tingling, your fingers start tingling, right? You might have a hyperactive nervous system, so like an alkalosis, whereas, you know, as you start to slip into a coma, right, in acidosis, you're going to have lowered excitability of neurons, and that's part of the problem. Okay, so those are observable. I did try to look up for you exactly what's happening in a neuron that makes this happen, and I couldn't find it. But if you guys can find it, if you're bored and want to look through scientific articles, I'd be super happy to know. Since we're also thinking about solutes in the kidneys as relate to our acid-base balance, it's important to note that the pH of your blood is in some ways linked to the potassium balance. So a thing that you would observe in blood is that in acidosis, so in people who have a blood pH of less than 7.35, they're also going to have high concentrations of potassium. So acidosis also causes hyperkalemia, whereas alkalosis causes the reverse, right? So if you have a pH of above 7.45, you're going to be in hypokalemia. So there's not going to be enough potassium. And this also kind of links into the symptoms that you would expect to see with each of these conditions. changing the amount of changing the amount of potassium in your blood kind of contributes to these effects that we see. Okay. Some of the effects that we would expect to see in terms of like observable things about the body would be things like cardiac arrhythmias and vasodilation. Okay. So here, what we're looking at is basically tracking the input and output of those hydrogen ions. So when we're thinking about blood pH, we're thinking about acidosis as being too many hydrogen ions and alkalosis as being not enough hydrogen ions. So it's important to kind of think about where those hydrogen ions are coming from, because this is kind of going to tell you about what the actual problem is, right? Like if you observe the blood pH, it's helpful to know that you're at the wrong pH and you need to fix it, but it's kind of more helpful to be able to think about what's causing the problem, whether it's behavioral, whether it's some organ in the body. What you really want to figure out is what the cause is, what part of the system is going wrong. So things that are going to put hydrogen ions into your blood come from the diet. So on the input side, protein metabolism and fat metabolism both create hydrogen ions in your blood, essentially. So they're both going to create acidity in your blood. For fats, that's possibly easier to remember, because remember we have fatty acids when we're thinking about lipids. So fatty acids are acids, which is why they contribute to making your blood acidic, which is essentially a high concentration of hydrogen ions. Proteins do the same thing. It's a little more complicated exactly what the acid is there, but the breakdown of proteins also increases acidity in your blood. There are also metabolic factors that are going to increase the concentration of hydrogen ions in the blood. So we talked on Monday about carbon dioxide and how carbon dioxide is really a biological acid because it can break down and give us hydrogen ions, essentially. Lactic acid and keto acids are also biological acids that would come from metabolism. So keto acids from the breakdown of proteins and fats, and lactic acid would come from basically exercise, right? So if you're exercising really hard, run out of oxygen, you move into lactic acid fermentation, you're going to end up with lactic acid in the blood, which is an acid, lowers the blood pH. So those metabolic factors and how hard your muscles are working, how hard your heart is working, what exactly you're breaking down for energy are all going to affect the pH of your blood. On the output side, we really only have two places we can get rid of hydrogen. One is the kidneys, which is what we're going to talk about today. We're going to actually directly secrete hydrogen into the tubular fluid and therefore pass it through the urine and excrete it that way. The other sort of output of hydrogen is going to be the lungs. What we're actually outputting in the lungs is carbon dioxide, but because carbon dioxide can basically be converted into hydrogen and hydrogen can get converted into carbon dioxide, it's basically the same thing from this perspective when we think about its interactions with carbonic anhydrase and bicarbonate, which we'll do more today. Okay, so acid-base disturbances in the blood, so messing with the pH of your blood, can come from respiratory disturbances, so either hyperventilating, so getting too much oxygen, not enough carbon dioxide staying in your blood because you're breathing it out too quickly, or the reverse, hypoventilation, not getting enough oxygen, and therefore having too much carbon dioxide because you're not breathing out fast enough. That's going to cause a change to your pH of your blood plasma because carbon dioxide is an acid in biological systems. And the reason it's an acid comes back to this equation that we saw a couple of times on Monday and also way back at some point in the respiratory system, we did look at this briefly, I believe. But basically, carbon dioxide mixed with water in the presence of carbonic anhydrase, which is basically an enzyme, creates carbonic acid, which then dissociates to bicarbonate and hydrogen. So this is why we can kind of say like carbon dioxide and our hydrogen ions are equivalent when we're talking about the contents of blood plasma, when we're talking about outputs and inputs of hydrogen ions. I recognize that there's not hydrogen in the chemical formula for carbon dioxide, but because this reaction is happening all the time in your body and there is always water in your body, from a functional perspective, they're substitutable because we can run this reaction in both directions. Okay, so remember that the normal concentration of carbon dioxide in your blood is 40 millimeters of mercury. So that's the normal partial pressure of carbon dioxide. And we tracked that through the body when we were talking about respiration. So that normal 40 millimeters of mercury is maintained by our respiratory reflexes. So if we change much from our normal partial pressure of carbon dioxide in our blood, that's going to cause your respiratory system to kind of compensate. So if your respiratory system is functioning appropriately, that'll balance itself back out if the problem is carbon dioxide. Unfortunately, your respiratory system may not be working appropriately, so sometimes that chemoreceptor reflex isn't going to work. Other sources of carbon dioxide include metabolism. So as you break down glucose, right, you're going to create carbon dioxide. So that's a place you could get carbon dioxide in your arterial blood. And the output is supposed to be through the alveoli, right? So since carbon dioxide is a biological acid, if we're increasing the plasma content of carbon dioxide, we're going to end up in what we call respiratory acidosis. And it's important here to note that it's respiratory acidosis because we're talking about carbon dioxide. We'll see that we also have metabolic acidosis, and we'll track through what that is as we go through the stuff today. If we have a decreased carbon dioxide in the blood plasma, we're going to be in respiratory alkalosis because a decrease of acid means your blood is getting more basic or more alkaline. So it's just the reverse. So just remember, lots of carbon dioxide, very acidic. Okay. So that's respiratory acidosis and alkalosis. When we talk about metabolic acidosis and alkalosis, what we're really going to track is directly those hydrogen ions, or on the flip side, bicarbonate ions. So remember that bicarbonate ions are actually kind of like a buffer for hydrogen ions. I'm going to flip back to the last slide for a second. So looking back at that equation, right, both of these pieces are reversible. So carbon dioxide makes your blood acidic, but one of the ways we can handle increases in acidity is if we have a lot of hydrogen ions, we bind them on to bicarbonate, create carbonic acid, which can then be converted into CO2 and H2O as well. So this bicarbonate acts as like a sponge for our hydrogen ions. So in metabolic acidosis, we can end up in acidosis either because we directly have more hydrogen ions, or because we're losing some of that sponge, some of that buffer. So those hydrogen ions that were in the plasma no longer have anywhere to go. They're not getting soaked up, so they're directly going to affect the pH at that point. So when we're talking about metabolic acidosis, we're talking about factors other than CO2, because CO2 can be regulated by regulating our breathing. But when we're changing our pH due to our hydrogen ions or our bicarbonate, while it's a related system, it's going to kind of require some different regulation there. So decreases in the pH could occur through a high protein or high fat diet. Those produce acids when we break them down. Heavy exercise can decrease our pH because when we run out of oxygen, we have to do something anaerobic, right, which produces lactic acid as a byproduct of glycolysis. Severe diarrhea can also cause metabolic acidosis. And this is because some of the bicarbonate is in your intestines, basically. So it's running through your digestive system. And if you have severe diarrhea, instead of having time to kind of like reabsorb bicarbonate or to absorb it from your food in the first place, you're going to excrete it. So you lose bicarbonate, which means you lose some of this buffer for hydrogen. Another place we can end up in metabolic acidosis is with kidney problems. So if you have renal dysfunction of some type, this can cause acidosis because your kidneys aren't able to appropriately balance the pH. We'll be looking at, of course, our cells along our nephron in our kidneys to look at how they balance pH normally, like when your kidneys are working appropriately. And hopefully as we go through that, it'll be kind of clear why, if the system breaks, you could end up in acidosis. We can also have alkalosis, so metabolic alkalosis. We can have respiratory acidosis, respiratory alkalosis. We can have metabolic acidosis, metabolic alkalosis. So metabolic alkalosis, your blood is going to become more basic, which means you're losing hydrogen ions, or on the flip side, you could be gaining bicarbonate ions. You have tons and tons of bicarbonate ions. You're going to bind up a bunch of those hydrogen ions, which is going to raise the pH of your plasma. So metabolic alkalosis is just when we're raising the pH through something other than our respiratory system, so through something other than carbon dioxide. So some examples of things that would cause metabolic alkalosis are vomiting. So one of the places you see tons and tons of hydrogen ions are in your stomach acids. So if you start vomiting out stomach acids, you're basically vomiting out hydrogen ions, which means you then have fewer hydrogen ions in your body. As you have fewer hydrogen ions in your body, you ultimately end up with fewer hydrogen ions in the blood. You can also increase pH through the consumption of alkaline products. So like if you eat baking soda, which I by accident ate too much baking soda, right? When you make beer bread, you put baking soda in there to make it rise. So if you consume alkaline products, if you're a bad baker like me, you can end up in metabolic alkalosis because you have too much bicarbonate, because baking soda is like just bicarbonate. Also, you can end up in metabolic alkalosis through renal dysfunction. So if any of the systems that we're going to look at next break, you can end up having too little hydrogen or too much bicarbonate. Okay. Your body has defenses against acidosis and alkalosis, as you would expect, because it's regulated, right? We want to be in homeostasis. So the first line of defense is just chemical buffers, right? So we can buffer hydrogen ions through bicarbonate, and we'll also see some phosphates. Buffering of those hydrogen ions can only limit changes in pH. It can't actually like reverse the problem. And it especially can't like make a huge difference if you're in extreme acidosis, because you only have a certain amount of buffer in the plasma, and at some point it's like going to be full. And those equations are reversible, so at a certain point it's not going to run in the direction you want. So it can't really reverse these changes unless you add more buffer. So we have two sort of longer term ways that we can affect the pH of the blood. So just like the kidneys and our respiratory system can mess up the pH of our plasma, they can also kind of fix it. So we can compensate through our respiratory system to reverse a change in pH. So if you are breathing in more oxygen than you're breathing out carbon dioxide, you can adjust how you're breathing, basically. So if you have too much carbon dioxide in your blood and therefore your blood is too acidic, you can just breathe out that carbon dioxide. So that would count as respiratory compensation. And you can do respiratory compensation for renal problems, right? Like, you don't just have to do respiratory compensation for a respiration problem. You can use it to control plasma pH overall, not just plasma pH as a result of like messed up carbon dioxide. Same deal with renal compensation. So your kidneys can change the pH and they can reverse changes in pH. So specifically what we're going to see that the kidneys do is that they secrete hydrogen into your renal tubules and therefore are able to excrete hydrogen through your urinary system. They can also reabsorb bicarbonate. So just like we reabsorb water, just like we reabsorb sodium, we can reabsorb bicarbonate and we can kind of upregulate that. They can also synthesize bicarbonate. So we'll see that they can actually kind of make their own bicarbonate. So that's a really kind of long-term effective way to change the pH. Likewise, and they can toggle this, right? So they can either increase hydrogen secretion and bicarbonate reabsorption and synthesis, both of which would affect too much acid in your plasma. So both of those would affect acidosis, or they can also fix alkalosis, right? So alkalosis would be a too low a concentration of hydrogen ions. So they could decrease hydrogen ion secretion, because hydrogen ion secretion is basically how we're dumping hydrogen, so we could conserve it. And at the same time, they would decrease the reabsorption of bicarbonate, because you wouldn't want to bind that hydrogen anymore because you're trying to keep it up. Okay. So the exact mechanisms of each of those three ways that we can defend against changes in pH. The first is just buffering, so chemical buffering. Now, the most important buffer that we really focus on, both because it's involved in the kidney function, so it's both going to be involved in our renal system, as well as the fact that it's one of our like primary byproducts when we're breaking carbon dioxide and water down ultimately to bicarbonate and hydrogen. So bicarbonate is a really important buffer, and it's the one that's most present in our extracellular fluid, so fluid outside the cytoplasm of the cells, so hanging out in the plasma, basically. So we got bicarbonate hanging out in our plasma. You see our bicarbonate. We bind bicarbonate onto hydrogen. So then we've created something that's not an acid anymore. Well, kind of is, but it's not free hydrogen ions. So we got our carbonic acid. Inside the cells, so in the intracellular fluid, we have other buffers. So inside cells, proteins can actually act as a buffer against changes in pH, because some of our proteins can bind hydrogen. So for example, we saw that like hemoglobin could bind hydrogen. Other proteins can also bind hydrogen. And phosphates can also bind hydrogen. So here we see, sorry guys. Okay, so here we see our phosphates binding on hydrogen, also acting as a chemical buffer. So these are set in order basically of how quickly they can adjust the pH. So using a buffer in the plasma or inside our cells is going to happen really quickly because it's just a chemical reaction. If we're trying to adjust our respiration or adjust our renal function, those are going to be slower. So our respiratory compensation is like the next fastest, but this is going to take a couple minutes. So buffering is almost instantaneous, almost automatic. But if we have a bigger problem than the amount of buffer in our plasma or in our cells can handle, we're going to need to change something through our organs. So the next quickest thing we can do about a messed up pH in our plasma is respiratory compensation. So we consider this our second line of defense, and it's going to regulate the pH by changing ventilation. So if we have blood that's too acidic, so we're in acidosis, either because we have too many hydrogen ions or because we have too much carbon dioxide, very simply, we can just increase ventilation. And increasing ventilation is going to fix acidosis, because if you breathe faster, you're breathing out more. And as you're breathing out more, this decreases the carbon dioxide in your blood. The reverse is also true. So in states of alkalosis, so you have too little hydrogen ions or too little carbon dioxide, because those are kind of interchangeable in our system, in your blood. So your blood pH is too high. You can decrease ventilation, so breathe slower. And this is going to maintain the carbon dioxide in your blood longer. It's going to get that concentration of carbon dioxide in your blood up, which is going to bring the pH of your plasma back down, partially because we can convert that carbon dioxide into hydrogen ions. So this takes a little bit of time, but still relatively quick compared to our renal mechanisms. Okay, so this is just a quick flow chart of that respiratory compensation and how it would work to affect acidosis. So just like usual with these diagrams, you can flip the arrows if we want to talk about alkalosis. So if we have a low pH, so here's our low pH, we're in acidosis because we don't have enough hydrogen ions in the blood, or enough carbon dioxide, because these are really interchangeable. It's going to be picked up by our chemoreceptors in, like in our cardiovascular system, basically. So our chemoreceptors are going to detect and respond to this blood pH. And the way that they're going to react is to increase ventilation. So they're going to increase ventilation in order to breathe off more carbon dioxide. As we breathe off more carbon dioxide, it means we're going to lower the carbon dioxide concentration of the blood, which means that we're essentially lowering the number of hydrogen ions in our blood. This raises the pH of our plasma and is a negative feedback loop, because as we raise the pH of the blood, it kind of stops the system, right? Like we're actually correcting the pH, which means that we don't need to run the rest of these actions if our pH is fine. Okay. Our last line of defense against acidosis and alkalosis is renal compensation. So our kidneys, sorry about the phone. Weird to have a landline still, but hey. Okay, I'm just gonna talk louder while the phone is on. Okay, so when we are adjusting pH using the kidneys, this is gonna take hours or possibly days in order to adjust pH. So this is really a long-term regulation. It's slow. That's why this is our third line of defense. If we can fix the pH with a buffer, great, we don't need to do this. If we can fix the pH by just changing how fast or slow we're breathing, also great. Don't need to mess with the function of our kidneys, although depending how fast or slow we have to breathe, that might separately be a problem. But our kidneys can really long-term change pH by changing what's happening at their apical and basolateral membranes. That should not come as a surprise. at this point in the unit. So our kidneys are able to regulate hydrogen ions and bicarbonate. So they're able to either increase the excretion of hydrogen ions, so you can get rid of hydrogen ions, meaning you're getting rid of some of the acidity in the blood, or you can, well, not really, and you can reabsorb bicarbonate, because bicarbonate is one of our buffers. Reabsorbing bicarbonate is going to bind on those hydrogen ions, which is going to take them out of the plasma for an amount of time. So that's also going to help correct our pH. A cool thing that the kidneys can do is that they can also make new bicarbonate in the renal tubules. So they don't just have to work with the bicarbonate that like you're eating or drinking. They can also make bicarbonate basically from carbon dioxide. So it's not that exciting, but we'll see how that works in a second. So that's partially just important to note, because like if the problem is diarrhea and that you're excreting too much bicarbonate, it wouldn't be helpful if you could only regulate this through conserving bicarbonate. So it is important that you can also synthesize bicarbonate. Okay, so in states of acidosis, we're going to see increased secretion of hydrogen ions. So if you have too much hydrogen in your blood, right, so your blood is too acidic, we're going to secrete hydrogen ions into the renal tubules and therefore into the urine. So remember that secretion basically ultimately causes elimination of whatever we're secreting, because secretion is going from the plasma into the fluid in your renal tubules, which is essentially urine. It's like future urine, right? In those same states of increased acidity, we're going to want to reabsorb bicarbonate so that we can buffer out that hydrogen in the plasma. We're also going to increase our synthesis of new bicarbonate, because while we're reabsorbing bicarbonate, if what we want is more bicarbonate, let's just make some. This is going to happen a little differently in the proximal tubules than in the distal tubules and collecting ducts. And we're actually going to see our second type of epithelial cell in the distal tubules and collecting ducts. So thus far, we've been talking about principal cells. So finally, we're actually going to see an intercalated cell, but not true of the proximal tubule, which we're going to do first. So in the proximal tubule, almost all of the bicarbonate that gets filtered from the glomerulus is going to get reabsorbed. So just like we're reabsorbing everything else, our proximal tubule is our mass reabsorber. We're going to reabsorb bicarbonate. And as we look at these cells, we'll see that bicarbonate reabsorption is coupled to hydrogen ion secretion. So as we reabsorb bicarbonate, we are also getting rid of hydrogen ions. So this is also going to be dependent on sodium. So we've talked before about how sodium really drives a lot of the reabsorption in our cells, because it was kind of driving reabsorption of water. We'll see that it's also linked into our system for reabsorbing bicarbonate and secreting hydrogen. When we get to the distal tubules, we're still going to be secreting hydrogen, but what we're also going to be doing is synthesizing new bicarbonate ions. And this is important because like by the time you get to the distal tubule, right, like you already reabsorbed 80 to 90% of your bicarbonate. So there's not really bicarbonate left by the time that fluid gets to the distal tubule and the collecting duct. So instead of reabsorbing more bicarbonate, your distal tubule and your collecting duct create new bicarbonate, which you can then pull into the plasma. So under normal conditions, the bicarbonate that you're making are going to get added to the plasma, and the hydrogen ions are going to get secreted into tubular fluid, just like in the proximal tubule. What we're going to see is a little different is that because there's no bicarbonate really in that fluid by the time we get to the distal tubule, we're going to use a different buffer for hydrogen ions. Specifically, we're going to be buffering those using phosphate ions. Okay, so first what we're looking at here is the proximal tubule. Okay, so in this proximal tubule epithelial cell, there's a lot to see. We can see there are a lot of transporters. We're going to talk about each of them, but we're going to start in the tubular fluid first. Okay, so what we had happening was our plasma, everything in the plasma is getting filtered into the tubular fluid. So this includes bicarbonate. So here we can see our bicarbonate. It's indicating that it was filtered. Hydrogen ions, it says secreted. We're going to look at the secretion here, but just know that there are also hydrogen ions in the tubular fluid. Okay, so we got our bicarbonate that we're filtering through our tubular fluid. We bind it on to our hydrogen ions, creating carbonic acid, which can then be converted by carbonic anhydrase here in the apical membrane. So we just have some carbonic acid, or sorry, some carbonic anhydrase in the apical membrane, like inserted in there. So it then converts this into water and CO2. CO2 can dissolve directly across the plasma membrane. So we see just diffusion of carbon dioxide into our epithelial cell here across the apical membrane once we've converted basically our bicarbonate and hydrogen into CO2. So the CO2 moves into our proximal tubule, into the epithelial cell, and this CO2 encounters more water that was in our cytoplasm. So we take that CO2, we grab some water, we have more carbonic anhydrase floating around in the cytoplasm of these proximal tubule epithelial cells. So we can, again, run this conversion, but in the reverse. So carbonic anhydrase converts it to carbonic acid, which can then dissociate into hydrogen ions and bicarbonate ions, because that's what it does. It just dissociates. So we had our hydrogen ions and our bicarbonate in the tubular fluid, converted them to water and CO2, absorbed the CO2, and then basically turned them back into hydrogen ions and bicarbonate. So that was kind of a complicated system just to get them into the cytoplasm, but we did that so we basically didn't have to use transporters and could do this by diffusion of carbon dioxide. So then we can track each of these pieces separately. So now we've got our hydrogen ions and we have our bicarbonate ions. What happens with the hydrogen ions is basically we have a pump that pumps out hydrogen ions. So that's going to be secretion of hydrogen ions into the tubular fluid. So this was just kind of a loop. This is why it says hydrogen ions secreted here, is because as we pump out those hydrogen ions, we can use them to reabsorb more bicarbonate, basically. So this could just be like a circle if what we're tracking are the hydrogen ions. We also are able to counter transport, so like using an antiporter, we can move hydrogen ions out of the cell while we reabsorb sodium into the cell. And we remember that we can move sodium into the cell down its gradient because our sodium-potassium pump is keeping sodium in our proximal tubule epithelial cells low. So that's what's going on with hydrogen. Both of these pieces, for hydrogen, we're seeing hydrogen secretion. So we're really just concerned with the apical membrane when we're talking about hydrogen being secreted in the proximal tubule. But at the same time as we're secreting hydrogen and therefore possibly peeing it out, right, if we're trying to get rid of hydrogen, we're also reabsorbing bicarbonate. So we use the same system to get that bicarbonate into the cell, right? We filtered in some bicarbonate, turned it into water and CO2, moved that CO2 into the cell, eventually convert it back into bicarbonate and hydrogen ions. Once we have this bicarbonate inside the cell, we're going to move it into the plasma. So when we're reabsorbing bicarbonate, we're going to be thinking about our basolateral membrane. And we have two types of transporters that we're interested in here. One is co-transport with sodium. So while we actively reabsorb sodium here, yeah, we just do. You can see this is active transport because of the red arrow. We pull along some bicarbonate with it through this type of transporter. So here we have our co-transport. And then we also have a counter-transport mechanism with our chloride ions. So sometimes we're going to be actively moving chloride ions into our tubule epithelial cells. And at the same time as we do that, we push our bicarbonate ions out of the cell. So that's another way we can reabsorb the bicarbonate ions. Okay. So just to summarize, because that was a kind of a lot of steps in here, or at least I feel like that's a lot of arrows. We have hydrogen ions being secreted through a hydrogen ion pump and through counter transport with sodium here on the apical membrane. And this has the end result of excreting hydrogen ions. So it would lower blood pH. So it would, yeah, it would lower blood pH, so like fixed alkalosis. And sorry, fixed acidosis. And sorry, it would raise blood pH. I'm sorry, guys. Whatever, we're getting rid of hydrogen ions, so we're becoming less acidic. That's what I meant to say. At the same time, we're pulling bicarbonate ions into the plasma, which act as a buffer for hydrogen ions. So it also removes hydrogen ions from the plasma. Essentially, as we remove hydrogen ions, we also become less acidic. Okay. When we get to the distal tubules and the collecting ducts, we're actually going to start talking about the creation of new bicarbonate ions. So this is important to note. This is now an intercalated cell. So that was our second type of cell in the tubules, the one that we haven't really talked about yet. So its job is to create more bicarbonate ions. So the way it's going to do that, basically, is it's soaking up carbon dioxide from the tubular fluid, binds it onto water, eventually creates hydrogen ions and bicarbonate. It can then absorb those bicarbonate ions that it just created through a chloride ion, bicarbonate ion countertransporter. So we saw this countertransporter was also present in our proximal tubule. What we can also see here that we didn't see in the proximal tubule is that we're basically going to recycle these chloride ions through a chloride ion channel. What we don't see in these intercalated cells in the distal tubule, flipping back, are our sodium bicarbonate co-transporters. So those are only present in the proximal tubule, not present in the distal tubule. We just have our chloride and bicarbonate countertransporter. As we made this bicarbonate, we also created hydrogen ions. We don't want those to get into our plasma because the whole point is that we're trying to fix a problem of acidosis, which is basically too many hydrogen ions. So we take those hydrogen ions and we push them out through a hydrogen ion pump, just like in our proximal tubule. Moving back again so that we see that, hydrogen ion pump. But our second transporter that we see in the distal tubule is different. So in the proximal tubule, we were counter-transporting hydrogen and sodium. In the distal tubule, we're counter-transporting hydrogen and potassium. So we said, like several slides back, that the acidosis and alkalosis are also related to potassium concentration in the blood. So this is part of where you might see that, right? Okay. Next to the distal tubule and the collecting duct are where we're going to see a different buffer in the tubular fluid. The reason we need a buffer in the tubular fluid is there's basically like a floor on how acidic you can make your urine. You can get down to a pH of 4.5, but if you get lower than that, basically these proteins are kind of going to shut off. So in order to get around that, we use this phosphate buffer in the tubular fluid to kind of control the pH of urine as well. And we have to do that with phosphate ions because we don't have bicarbonate there anymore. Okay, so we are out of time, so we will pick up with this later. We don't have class on Friday because technically it is Easter break, although in these weird times that seems strange. But if you celebrate Good Friday, have a Good Friday. If you don't, just have a regular Good Friday. So I'll see you guys later.